My name is Jace Duhan, and I'll be presenting on identifying heavy metal decontaminating bacteria from plants and Superfund sites. This was done as part of my coursework for genetics lab at the University of Alabama at Birmingham. So what are Superfund sites? So essentially, these are areas of extreme environmental contamination, which the EPA has set aside to try and clean up the hazardous materials present in these locations. So those hazardous materials could be, asbestos or radioactive material, or what we'll specifically be looking at is heavy metals. Um, so this was the Superfund site that we collected samples from. Um, so this is the one in Birmingham, Alabama, and it's on 35th Avenue. Um, so basically, we wanted to look at the heavy metal effect on plants. So we collected plant samples, and essentially this is a diagram showing that these heavy metals like cadmium and chromium, copper, lead, zinc, nickel, um, these heavy metals have detrimental effects on plants, which are toxic. They can trigger oxidative stress, necrosis, disrupt photosynthetic machinery so that the plant can't get energy, um, modulate their antioxidant systems in a negative way, and really change a lot of biochemical pathways that are important for plants to survive and thrive. So essentially, bacteria can actually help these plants process heavy metals. So bacteria inside the plant's, I guess, natural microbiome, sometimes can help process these heavy metals. And we see, on the left here, there's a plant that doesn't have the bacteria helping out, and then on the right we have this bacteria helping out. And we can see the plant on the right is much healthier. So I wanted to look at, are there heavy metals processing bacteria associated with plants living in these Superfund sites that maybe help these plants grow in this harsh environment? And so the way that happened, the way we wanted to look at that was there were samples collected from the Birmingham Superfund site. Essentially, the samples were processed to try and kill the bacteria on the outside of the plant, but preserve the bacteria on the inside of the plant and have access to it. So then those microbes were spread after LB media was prepped. They're cultured, and then they were isolated, and the 12 bacteria were selected. Then their DNA were isolated, they were replicated, and then there was Sanger sequencing done to try and identify the identity of these bacteria. So here are the two plants that were collected. The first is matted sand mat, and the second is Brazilian vervain. These are the two plants that I isolated the bacteria from. And these are the 12 bacteria I ended up selecting, and this is them isolated on the street plate. So there are 12 bacteria, and then of those 12 bacteria, each of their DNA was isolated using a procedure similar to this. And then the DNA was amplified using PCR. So forward and reverse primers were selected. The DNA was replicated, and then it was sequenced using Sanger sequencing. So essentially what I found was that I had these 12 bacteria, and 6 of which were actually related to heavy metal contamination. So we can see here, 1, 2, 3, 4, 5, 6. These are the different strains of bacteria. And so essentially, since 6 out of 12 bacteria identified were associated with this decontamination of heavy metals, and actually 5 of those related to plants and soil, so this kind of suggests a strong correlation between how these bacteria are associated with the successful growth of plants in these hazardous sites or these heavy metal rich sites. But of course, more research needs to be done on exactly how these bacteria will impact the growth and development of the plants, or what role they're really playing in this in these Superfund sites and these plants. So essentially, here are my references. Thank you.